So we just finished up our organic chemistry type tonight. So that was the first little bit. In class, we been kind of ping ponging back and forth between these. So I would finish carbohydrates and then go look at this sheet here, which is the diagram notes. I'm going to run through all of them on this one little video for you guys. So 4 types of organic macromolecules. The first one that you see here are your carbohydrates. This basically says everything in the type notes. I'm more of a diagram person. I like things picture rather than just all the words there. So it is the same info. If you need more help, please look at this, or if you just want to help solidify the information you have, please watch this. Carbohydrates, carbon, hydrogen, and oxygen. I put those little ratio. You always have a 2-to-1 ratio with hydrogens to oxygens. Usually I'll say like, oh, your carbohydrates are in a 1 to 2-to-1 ratio. For every carbon, 2 hydrogens to 1 oxygen. We know as you get larger and larger disaccharides, polysaccharides, not always the case. However, you will always have a 2-to-1 ratio with hydrogens and oxygens because that's what we're kind of pulling out in order to go through dehydration synthesis to join these smaller molecules together to create your large carbohydrates. We know that they are your energy sources within your body. You have 3 types of carbs: monosaccharides, disaccharides, and polysaccharides. Monosaccharides meaning one sugar. This is your quick energy source within your body. It's the first place your body wants to go to burn in order to create your ATP or energy. The diagrams that you see here, I will never go on a test. Oh, let's look at this diagram. Is that glucose, galactose, or fructose? So that's not going to happen on a test. I might show you one of these diagrams and say, am I looking at a carbohydrate, a lipid, a protein, or nucleic acid? You go, oh, it's a ring. Sugars usually are going to be in rings. You can also look at your elements: carbon, hydrogen, oxygen. 1 to 2-to-1 ratio. It has to be a sugar there. So those simple sugars are called your monosaccharides. They all have the same formula, C6H12O6, but if you look at them, they're all structurally a little bit different. Fructose, a 5-sided sugar. These are my 6-sided sugars. The only difference between glucose and galactose is the location of that hydroxyl group. Here it's facing up, here it's facing down. It actually makes it a slightly different sugar. But all 3 of those will have 6 carbons, 12 hydrogens, and 6 oxygens, which is why they are called isomers. Same formula, different structure. I can take those particular monosaccharides and join them together. It's always glucose plus something. And when I do that, I create disaccharides. It will be created by dehydration synthesis. I am removing water. I am bonding things together. So when I join 2 molecules, here it is, glucose and glucose, C6H12O6 plus C6H12O6. I yield a molecule. You think it should be C12H22O, or I'm sorry, C12H24O12, but it's not. It's not exactly double. And the reason is you need to take out water. You need to take out 2 hydrogens and 1 oxygen. So if I join 2 monosaccharides together, my formula will always be C12H22O11, and then show that you took out that H2O there. That way your formula will be balanced on both sides. So here I have glucose and glucose. I am taking out my little hydroxyl group off of one and a hydrogen off the other. It does not matter which end you actually will lose that off of. But what you're left with is a bonding spot here, which is your oxygen. So now this is storing energy because we created a bond. Potential energy is stored within that bond. So glucose plus glucose, monosaccharide plus monosaccharide yields maltose. This maltose is a disaccharide. I'm showing my water here. If I reverse that arrow and I go in the blue direction, now I have hydrolysis. So I'm adding water to this big molecule, basic digestion, catabolic reaction. I'm breaking that bond. I'm restoring these particular endings here, and I end up with 2 smaller monosaccharides. I am now releasing energy when I do that because I broke a bond. Here are my other disaccharides: maltose, sucrose, lactose. We just looked at maltose. It's always going to be glucose plus something. So glucose and glucose is maltose. Glucose and fructose is sucrose. Glucose and galactose is lactose. Just like glucose, galactose, and fructose are isomers, so are my disaccharides: maltose, sucrose, and lactose. C12, H22, O11. If you draw them out, they are all different from one another. Your polysaccharides, huge molecules. They're used for structural, they're used for storage within the particular organisms. So if you are a plant, for these many sugars joined together, you can store those excess glucose molecules as starch. So think like potatoes, they have these little starch granules that they store their excess food in. They'll utilize that later. They'll break it down for cell respiration so that they can grow, so they can repair, so they can do all those activities. They can store it as amyopectin, and we talked about pectin being that thickening agent for those jams and jellies. If you know anybody who actually creates jams and jellies, but it thickens up that fruit so that it settles. It can also be structural. So structural polysaccharides within plants is cellulose, and cellulose is what makes up the cell walls within those particular plants. So remember, humans don't have cell walls, so we do not have cellulose within our cells. The way that our bodies store our excess glucose molecules is it strings them all together into a very large polysaccharide and stores it in the liver. That is our sugar storage for as we need it. So let's say you are that marathon runner, or even our sports teams. You guys have your pasta meals, okay? Pasta dinners like before a big match, or you're eating a ton of pasta before you go out and run a marathon. And the reason is because you want all that glycogen, those sugar molecules, because we know pasta is a lot of sugar once you break it down, starch. We take it, we store it within our liver so that it can be broken down slowly over time as we need that energy. So I always say, if you're a marathon runner, do you want to eat a lot of pasta, huge polysaccharides that we can break and break bonds and get energy from? Or do we want to eat like a Milky Way or a Snickers bar, lots, a very small molecule, very little energy, not a lot of bonds, not going to last you guys a long time. So of course we want to eat that pasta or that starch or those larger sugars that we have. Insects and fungi. They have chitin, which is a structural polysaccharide. Within fungus, it makes up the cell walls. Within your insects, arthropods, it makes up their exoskeleton. Next, we move on to your lipids. Same thing as carbohydrates, carbon, hydrogen, oxygen. But here we have very few oxygens. So your formula is nowhere near a 1 to 2 to 1 ratio. And the reason is because you have these really long hydrocarbon chains that you guys are going to see. When we go to create a lipid, it is a glycerol molecule. You'll see it here. That is the backbone of my lipid, C3H8O3. You'll see it on your flashcards. And to that, I'm going to add 3 fatty acid chains, which is what I'm looking at here. This is one example of one. Fatty acid chain is just a hydrocarbon chain, hydrogens and carbons, that's it, with a carboxyl functional group on the end. That was the first set of notes we looked at. It's carbon, double bonded oxygen with an OH group. That is my carboxyl group. Notice here on my hydrocarbon chain, dot, dot, dot. So I'm bonding one carbon to another carbon with this dotted line. What that means is that in between those dots, I might have another 400 carbons. I might have another 50 carbons bonded around hydrogens. It's just saying this is what the end of my chain looks like. This is what the front of my chain looks like. This hydrocarbon chain can vary in length, and that's what that dot, dot, dot basically is representing. So the length of the chain is going to give me a different fat. Also, the number of bonds in between those carbons is also going to change my fat. And what I mean by that, we know that every carbon has to have 4 bonds around it. Well, if those bonds are single bonds, such as what we see here, single lines between them, that means I'm connecting this carbon to this carbon with sharing a pair of electrons. That is known as a saturated fatty acid chain. So we have all single bonds between carbons. So notice, this does not count carbon to oxygen. That's a carboxyl group. Yes, that is a double bond. Yes, they are sharing 2 pairs of electrons. It is not between carbons, it's between carbon and oxygen. In order for it to be saturated, we need all single bonds between carbons. So this is a saturated fatty acid chain that we're looking at. Those saturated fatty acid saturated fatty acids are not good for you. They are going to be solid at room temperature. They're usually coming from animal products, lard, butter, things that don't melt at room temperature. If you think like a steak with that big layer of fat around it, they are not good for you. They can lead to arteriosclerosis. They build up in the arteries, hardening of the arteries. It can lead to heart attack as well as stroke. We kind of switched from using lard and butter, kind of old school cooking, to using more of your unsaturated fatty acids to cook with. And that's more plant-based material. They do become liquid at room temperature. It's more your oils. So your unsaturated fatty acid, you have to have at least one double or triple bond between carbons. So here's my saturated, single bond, single bond, single bond. I have my carbons, single lines between them. I've got all my hydrogens around it. Each carbon's got 4 bonds to it. That is my unhealthy saturated fat. On the end of it is my carboxyl group. So we're once again looking at this hydrocarbon chain. With my unsaturated fatty acid chains, these are my healthy ones. You're going to go, wow, that looks a lot different than the one above. There's a double bond, double bond, double bond, double bond, double bond. And I have some single bonds here. Every place I have a double bond, notice I'm lacking hydrogens. There's no place for hydrogens to bond. Carbon still has 4 things around it. Let's look at this one right here. He's double bonded to this carbon, so he's sharing 1, 2, 3, 4. We have 4 electrons there. And the other side, we still have 4 electrons. He's got 8 electrons in that outermost ring, 4 of his own. And then he's sharing 2 here, and he's sharing 2 with this one here. So there is no place along the way for hydrogens to be able to fit through. This I can have a hydrogen to. This particular carbon, double bonded. He's got 4 of his own, sharing 5, sharing 6 with that carbon, 7 with this hydrogen, 8 with this carbon. So he has 8 in his outer ring. Anytime you see at least one double or triple bond between carbons in that hydrocarbon chain, once again, that carboxyl group does not count. We say that that is an unsaturated fat. That is your healthier ones, plant-based. They are liquid at room temperature. Just like we created a disaccharide, we can create a lipid. So here's your synthesis of it, dehydration synthesis. Here's my glycerol molecule, C3H8O3. Excuse me. To that, I want to add different fatty acid chains. So once again, dotted lines. Each of these chains can be different lengths. Some can be saturated, some can be unsaturated. So I don't want to take up the whole page putting hydrocarbon chains on it. So I'm saying here's what the first chain looks like. The middle could vary. To this glycerol molecule, I'm adding these 3 fatty acid chains, which means if I'm adding 3 things, I need 3 bonds. I need to take out 3 waters. Okay, so I'm losing a hydroxyl off one end, a hydrogen off the other. Doesn't matter who loses what. If I'm going to take the hydroxyl off my glycerol molecule and a hydrogen off here, that's fine. I'm left with an oxygen. This is my new bonding spot right here, right here, right here. Every place I have that new bonding spot, every place I added a fatty acid chain onto, I've taken out a water. Lipids are huge molecules. So my building blocks are glycerol and 3 fatty acid chains. Since I have 3 bonds, these lipids are actually going to provide more energy than your carbohydrates. So it is your secondary energy source where your carbs are your first place. Body burns all your carbohydrates off, burns the glycogen within your liver. It's going to your lipids next. Think about bears that hibernate for the winter. They're burning off that glucose and glycogen right away, probably before they even get to that hibernation state. Then throughout the winter, even though they're sleeping and everything's kind of slowed down, they still have reactions going on. They still have blood moving. They still have everything that needs to occur within their body. They're burning off their lipids. So they come out from hibernation and those bears are all thin again. And they have to work on next season building that layer of lipids back up. Otherwise, they're not going to survive that hibernation. Once again, I can go the other way. I can take this giant lipid. Let's say I give you a potato chip to eat, lots of fat in that. Put it in your mouth. You add some water to it, and you break it down to glycerol and 3 fatty acids and some energy there. Within our handwritten notes, that will be next for you guys. You guys will see glycolipids. What's the difference? Well, let's take off, instead of putting this fatty acid chain on, let's put a sugar on. Put a single ring sugar there. Glycolipids are going to help with nerve impulses. Make a phospholipid. Still glycerol, still 2 fatty acid chains. Instead of the third one, let's put a phosphate group, PO4 group there. So it makes up your cell membranes. Some of your steroids and hormones are lipid-based. So we'll talk about cholesterol and steroid nucleus. You guys saw that in the typewritten notes. You'll see it in the next set of notes as well. Hormones, testosterone and estrogen and cholesterol are the ones that we were looking at there. Proteins, carbon, hydrogen, oxygen, and now nitrogen. Sometimes you'll see sulfur, sometimes you'll see phosphorus, depending upon what of those 20 different amino acids you have. If we look at our basic structure of a protein is made up of amino acids. We have 20 different amino acids. We can string them together in super long chains and then kind of fold them and twist them and do all these different things to them to make different structures. But by doing that, we can create unlimited number of proteins. So by putting together those 20 different amino acids, we create unlimited number of proteins. Your smallest protein is about 50 amino acids in length. Your amino acids all have the same structure to them. So here's what we have. This is my amino acid. I have a central carbon. Once again, carbon's got 4 things bonded to it. Around that central carbon, I'm coming off, I have a carboxyl group. Carbon double bonded to oxygen with an OH. Same thing we saw on the end of the fatty acid chain. Going back to my central carbon, the other ending is my amino group. There's my nitrogen. That's how I know I'm dealing with an amino acid and protein. Going back to my central carbon, I have a hydrogen coming off of it. I don't care if the hydrogen's facing up or facing down. It doesn't really matter. The other ending off my central carbon is an R group. There is no element R that we're looking for. That R is a variable. It says Whatever is there is one of 20 different endings. Everything else besides that R group is going to be identical in every 20 of the amino acids. That R group could be a hydrogen. That's a specific amino acid. It could be a carbon with hydrogens around it. That's another amino acid. Maybe it's got some phosphorus in it. It's another amino acid. Maybe it's got some sulfur, different amino acid. But that R group is the only part of that structure that is going to change. I can take these particular amino acids and start putting them together. So if I take amino acid number 1, once again, I don't really care what it is. I'm just putting an R group there and join it with amino acid number 2. Dehydration synthesis, taking small things, making larger ones. Same exact process. Lose a hydroxyl off of one, lose a hydrogen off of the other. So I'm losing that hydroxyl off that carboxyl ending. I'm losing a hydrogen off the amino ending. H2O comes out. There's my new bonding spot that I have. So now I'm creating this new bond, and I end up with what is known as a dipeptide. 2 amino acids joined together is a di meaning 2, so it's a dipeptide. The bond that is joining them is called a peptide bond. So here's my dipeptide joined by my peptide bond, and I'm showing my water at the end. If I keep adding amino acids, let's say I add another one onto this dipeptide, take out another water, I now have a polypeptide. Another name for polypeptide is protein. Many amino acids join together. That's what a protein is. Proteins can be structural. They can help with transport. They can be enzymes. Huge section we'll be seeing. They can be toxins. They can be actin and myosin bands within your muscles. So there's a whole bunch that we're going to end up talking about as we go through. You guys also saw them typewritten notes. Last bit, nucleic acids, DNA and RNA, carbon, hydrogen, oxygen. Remember, if you're organic, you need to contain the element carbon. There's always kind of exceptions to the rule. I'm also going to have some phosphorus here, so that's that key element that we're going to see. So if you remember from first year bio, 1 sugar, 1 phosphate, 1 nitrogen base. Those are the parts that are going to make up my nucleic acids. So nucleic acids, deoxyribonucleic acid, ribonucleic acid, DNA and RNA are made up by what is known as nucleotides. So just like amino acids are your building blocks of proteins, our nucleotides are building blocks of nucleic acids. And what a nucleotide is 1 sugar, notice it's a ring, 1 phosphate, 1 nitrogen base. Their structure, when I string them all together, make up DNA or RNA. DNA, RNA, as well as ATP and GTP, adenosine triphosphate, and we have guanine triphosphate. These are other energy uses, or another energy use besides ATP, the energy use within your body. Your sugar in DNA is deoxyribose. So there's another way to realize that it's a sugar. First off, it ends in OSE. Second off, it is in a ring structure. So we talked about with our carbohydrates and monosaccharides. Ribose is the sugar that you will find in RNA. The only difference between deoxyribose and ribose and your glucose and galactose is a 6-carbon sugar, C6H12O6. Deoxyribose and ribose only have 5 carbons to them, so slightly different there. The big changes here is your DNA and RNA. If you guys remember DNA, deoxyribonucleic acid, it is a larger molecule. It is double-stranded. It is only found in the nucleus if you're a eukaryotic organism. It can't get out of the nucleus, but the DNA codes for all of your proteins, and proteins can only be made in the nucleus. So that's where RNA is going to come into play. So RNA is kind of like DNA's little brother. It's a single-stranded molecule. It's smaller. It can fit out through the nuclear pores. So my base pairs in DNA, adenine, thymine, guanine, and cytosine. So there's nitrogen bases, adenine, guanine, thymine, and cytosine. You will always pair adenine and thymine together. You'll always pair cytosine and guanine together. So you're like, well, why are there some lines between there, and why are they all highlighted? Because it kind of helps us know which ones go together and why. So why does adenine and thymine have to go together? In our regular notes, we talked about purines and pyrimidines. Purines, double ring structures. They're larger. So remember, these nitrogen bases are making up the rungs of our ladder. Adenine is a double ring structure. It is larger. You always pair 1 purine with 1 pyrimidine. Double ring structure. Adenine pairs with thymine, just like cytosine and guanine. So adenine and guanine are double ring structures. They are purines. Thymine and cytosine are single ring structures. You always pair one purine with one pyrimidine. So why can't adenine, my purine, go with cytosine, which is my pyrimidine? That's right, one purine, one pyrimidine. The reason is the number of hydrogen bonds between them. Adenine and thymine only have two hydrogen bonds. Guanine and cytosine have three hydrogen bonds. So it's the only possible way they can fit together when you are creating those rungs of the ladder. RNA has the same base pairs except one. Thymine gets replaced by what is known as uracil. It's everything that thymine is. So where we said thymine is a pyrimidine, a single ring structure, it's smaller, so is uracil. When it has two bonding spots to bond with adenine, so does uracil. So when we get to RNA, thymine gets replaced by uracil. So adenine pairs with uracil with two hydrogen bonds. Cytosine still pairs with guanine, still three hydrogen bonds between it. Notice that the phosphate group, there are high energy bonds between that and the sugar. So another flashcard thing. Don't just draw me PO4 with a little wavy line on the back of your structure. If you're going to draw me the structure of DNA, tell me where that high phosphate bond is. You need a little pointing area to there. We will do a huge section on nucleic acid. So it's like, ooh, I remember some of that, but we're just going over it real quick. We are. They're going to have a huge section. We'll talk about how it's done. We'll look at the whole process of protein synthesis. Remember in our notes, we also went through your three types of RNA, mRNA, tRNA, and rRNA, messenger, ribosomal, as well as transfer RNA. We kind of talked about what they did within those notes. So if you're not sure, go back to them, but we will absolutely touch base on them a little bit more when we get to our DNA section.